So here we have a student investigating the effect, this is question 5, of different sugar solutions on potato tissue. Quite a similar method to the one that we actually used in class. Okay, you've got all of your concentrations here and your results. Calculate value X in the table, which is a percentage change. Now, we did this before. We're actually looking at how much change in mass is in proportion to, with any percentage change, it's that original mass. Okay? And because it's a percentage change, you've got to times it by 100. Okay, it's always a proportion of the original value if it's a percentage change. So your percentage change is 11.31%, and it's a positive value because it's gained mass. Okay, why did the student calculate the percentage change in mass as well as the change in grams? The main thing is, if you have a look at theirs, just like with ours, they all had different starting masses. So they needed to make the results comparative, given that their starting mass was different every time. So it needed to be proportional. So then, girls, what you're doing is you're filling out a graph. We've done a graph similar to this before. Okay, if you do, I've put this on purpose, if you do a plot that you're not happy with, really with plots, you can do the plots in pencil, but everything else should probably be done in pen. Make it really clear that is not a plot. Okay, don't go mad with it, but just make it really clear. So I've put all my plots on there. I've put my values and I've labelled this as well. So you get 1 mark for putting a correct scale in here. Yours might be slightly different to mine, but that seemed to work quite well. It's got to be labelled as well with its correct unit, like I've done. The points need to be plotted correctly. They will check that, okay? And the line of best fit should accommodate half on each side of the line, as you well know, okay? Now, with this particular, you get 2 for the points by the looks of it. So just have a look at yours, check that you're all right with that. I've written here, error carried forward if the last question is wrong. So if you had calculated your percentage change wrong, you're not going to get penalised again for that, all right? You're going to get the full marks here. So, unsurprisingly, it's then asking you to use your graph to estimate the concentration of the solution inside the potato cells. So on my graph, I'm interested in this point here, because this is where no water enters the cell or leaves it, which means that the concentration inside those potato cells must be the same as the concentration outside. So no osmosis will occur. So therefore, I can read off of my thing that there is a value of 0.5 moles per decimetre cubed. Okay. The results in table 2 show the percentage change in mass of the potato cylinders. Explain why the percentage change results are positive and negative. So I've laid this out very clearly, okay? I've used my values from 0.0 to 0.4 millimoles per decimetre cubed, okay? Water went into the cell by osmosis. From 0.6 to 0.8, water leaves the cell by osmosis. Okay, so you don't necessarily have to go into that much depth, but it's just showing the examiner that you know that water moves in if there's more water outside, and it moves out if there's less water outside. Suggest 2 possible sources of error in the method given. So, where could it have gone wrong? The concentrations of solutions, we don't know whether they were exactly right. Whether the chips were dried properly in each case, whether the balance was accurate, whether there was any evaporation from the potato tubes. Question 6. Figure 4 shows parts of the digestive system. Label the stomach and the pancreas. Hopefully you all got that right. Okay, so just have a look. You had to get both of them for 1 mark. Okay, so many people suffer from stomach ulcers caused by a species of bacteria. The stomach is lined with a protective lining of mucus. Helicobacter pylori are acid-tolerant bacteria which can damage the mucus lining. Suggest how an infection with this bacteria might result in the stomach ulcer developing. So what that means is the bacteria is not killed by the hydrochloric acid. And so the lining, the mucus lining, is damaged. So the hydrochloric acid therefore damages stomach tissue, causing an ulcer. Okay, so you've got this idea that there's no protective covering anymore. Helicobacter pylori can also cause stomach cancer. Describe how a person is infected could also develop liver cancer. So presumably it's asking you to state how it spreads. So if the cancer is malignant, then the cancer cells can spread to other organs. And this normally happens via the blood, forming a secondary tumour. 6.4. Gluten is a form of protein found in some grains. Describe the test you would use to find out if protein is present in foods. So the protein test, you've got to add Biarritz reagent to the food sample. Now, Biarritz solution is, you will have used this in class, sodium hydroxide plus copper sulfate. Now, if you'd have said that instead of Biarritz, then that's fine. It's just easier to learn that it's called Biarritz reagent. If it turns purple, then protein is present. 6.5. Celiac disease is a disease of the digestive system. It damages the lining of the small intestine when foods that contain gluten are eaten. When people with celiac disease eat foods that contain gluten, 1, their immune system forms antibodies. 2, these antibodies attack the lining of the small intestine. And 3, this causes inflammation. Symptoms of celiac disease include poor growth. Suggest why a person with celiac disease might have this symptom. So, damaged villi will reduce the surface area for the absorption of food molecules. So you've got to get that whole idea that damaged villi, those villi, if you remember, look, if I do it over here. So that's the lining of the small intestine. The villi are the projections that normally increase the surface area so that food can be absorbed into the blood system. Okay. So fewer amino acids and glucose will be absorbed. This means there's less energy released from respiration. This also means that fewer proteins are going to be remade because there's less amino acids for growth. There's not much room for error there. Okay, you have to get all of these points. Okay, question 7. A gardener is looking at plants in his greenhouse. Some of the plants have a disease. Give 2 ways the gardener could identify the pathogen in the plants. So, look at a gardening manual, see if there's any pictures that they can compare them to, or send them to a lab for testing. Plants can become unhealthy if they do not have essential mineral ions. If there's a nitrate deficiency, they could have stunted growth. Nitrate helps them to make those essential amino acids if they don't have them. Magnesium deficiency, that means they can't make chlorophyll, so they get yellowing of leaves. Okay, plants need other mineral ions. Potassium ions are needed for healthy growth. Phosphate ions are needed for healthy flowers and fruits. The gardener makes his own garden compost. The percentage of minerals in his compost was compared to the 2 fertilisers he could buy. So, the gardener buys fertiliser S. Explain why he chose it. So we've got to make it clear that S, firstly, had the most nitrogen for healthy growth. There it is. Most nitrogen for healthy growth. Had the highest potassium for healthy roots or minerals. They've told you that at the top. Also, other information we could put down there. Again, look at those ticks. It's cheaper than tea. If we have a look. Yep, there it is. Cheaper than tea. Okay. It could be that flowers and fruits are less important for him. So it could be, for example, that he's growing a leaf like lettuce or cabbage or something like that, and therefore isn't that bothered about the fact that there's less phosphate in there. So that one was quite difficult to get, that last one.